Yeah, shall we start the class? Yeah, good evening to all of you. Shall we start the class? Smart. Ahmed, can I proceed with the class, Ahmed? Okay. Okay, so am I audible? Am I audible to all of you? No, Bajib. Am I audible? Okay, so medical parasitology. So you all are very much aware about what is parasitology, about the human parasites that infect in such a way that you have to understand the what do I say? First of all, what is a parasite? What is a endoparasite? What is a ectoparasite? Okay, the parasite that lives on the external surface of an organism is called as an ectoparasite. A parasite that lives within another organism is called as an endoparasite. Then a protozoa is something which is a unicellular organism like Plasmodium virus, Plasmodium vivax. And metazoa is a multicellular organism like helminths, worms, arthropods are all metazoas. And the last session we have already discussed about what is the eukaryote and what is the prokaryote. Then you know what is the host. A host is an organism on which the parasite lives. A definitive host is an organism in which an adult or a sexually mature stage of a parasite happens or lives. A definitive host is a host, basically an adult or a human, in which the parasite can undergo a sexual cycle. An intermediate host is an organism in which a parasite lives during its period of only the development. Zoonosis is a parasitic disease in which an animal is normally, or in which an animal is normally the host, but which also infects a man. Like corona causes COVID-19. COVID-19 is not a zoonotic disease. It's a human disease. It's not a zoonotic disease. It doesn't transmit through animals. The parasite doesn't, or the virus doesn't have the cycle into the, I mean to say in the animal. Then a vector is a living carrier that transports a pathogenic organism from an infected to a non-infected host. Initially, like you all know that female Anopheles mosquito is a vector for malaria, right? Similarly, there was also a presumption that your bats are a vector for coronavirus, but that's not true maybe. So that's how that is a vector. Vector is a carrier. Zoonosis, you know very well. So I hope all of you are clear. This is only the introduction to medical parasitology. We are not going to go in detail with the parasitology. It's not important for you, but you should know the definitions. You should understand what is a host, what is an intermediate host, what is zoonosis, what is protozoa, metazoa, endoparas, all the terminologies. I hope it is clear to all of you. Clear? Okay. So now we will discuss burden of some major parasitic infections. We start from Plasmodium. Plasmodium causes a disease called as malaria. Then you have a soil transmitted helminths, like you have got a roundworm which is called as Ascaris. You have got a whipworm which is called as Trichuris. You have got a hookworm which is called as Ankylostoma. Then you have got Scysiostoma. It causes disease of the renal system. Then you have got Filaria, which causes lymphatic, Filariasis or Elephantiasis. Then you have got Trypanosoma. Then you have got African trypanosomas, which is called African sleeping sickness. Then you have got Leishmania donovani or Leishmania, which causes your mucocutaneous and visceral Leishmaniasis. So these are the various parasites from Plasmodium to Filaria to Trypanosoma and Leishmania. Now the taxonomic classification of a parasitic organism. Now there is no universal accepted system for classification of parasites, and parasites form an important part of animal kingdom which comprises almost 8 lakhs identified species which are categorized into 33 phyla. The parasitic organisms that are of importance for human health are eukaryotes, and parasites are broadly classified into protozoa and metazoa. Protozoans or unicellular parasites have their own different peculiar morphology and means of locomotion, and there are 45,000 protozoan species. Metazoas include worms like helminths and arthropodas, which possess an external skeleton like lice and ticks. You have to note that the genus starts with a capital and species is always in italics. I hope you all know that. What is, like, for an example of Plasmodium falciparum. So you know the Plasmodium is the genus which always starts from caps. Okay. Now we will go into a little detail of the taxonomic classification of protozoa. Now the protozoa is belonging to a subkingdom which has got a phylum which is divided into multiple phylums, which is not important for you. Then you have got subphylum, then you have got genus. You should know the genus and the species. Entamoeba, Entamoeba histolytica, Giardia lamblia, Plasmodium falciparum, vivax, malaria and ovale. Balantidium is Balantidium coli, and Enterocytozoa is E. benusi. Examples of some of the intestinal protozoas which are transmitted by feco-oral route and cause diarrhea. Example is Giardia lamblia, is a worldwide parasite which has got its own habitat in a small intestine and results in malabsorption. Entamoeba histolytica invades the colon, causes bloody diarrhea, and that bloody diarrhea caused by Entamoeba is called as amoebic dysentery. There is a very important disease which is caused by Entamoeba histolytica, which is called as an extra-intestinal disease, which is called as amoebic liver abscess. Please remember that amoebic liver abscess is caused by Entamoeba histolytica. Then you have Cryptosporidium parvum, which is again prevalent in immunocompromised people. Then you have got Cyclospora cyatenesis. Then you have got Balantidium coli. It's a motile ciliated parasite which lives in the colon of pigs, humans, and rodents and can lead to colonic ulceration. Okay. So Entamoeba histolytica, which is very popular parasite, it causes typical flask-shaped ulcer in the colonic mucosa. This is a very important question. Flask-shaped ulcers caused by which parasite? Answer is Entamoeba histolytica. Examples of some systemic protozoas like Plasmodium. Plasmodium causes malaria. You have got 4 species. Toxoplasma gondii is transmitted by ingestion of oocytes from cat feces. Infection can lead to ocular issues. Leishmania transmitted by sandflies can lead to visceral leishmaniasis or cutaneous leishmaniasis. Then you have got trypanosoma, which is called as hemoflagellates, which causes in Africa sleeping sickness, and South America it is transmitted by bugs and leads to Chagas disease. Then we have got helminths, which are worms, which belong to metazoa. You have got nematodes. You have got roundworm, which appear round in cross section, and they cause. You have got 2 types of roundworms, especially when you go to worms in nematodes. Ascariasis or Ascarias is a roundworm. Trichuris is a whipworm. Ankylostoma is a hookworm. Necator is a hookworm. Enterobius is a pinworm or a threadworm, and strongyloids. These are all the, see, you have to just know the names. The total microbiology has got no concepts. There is no concept to explain in microbiology. It is just the names. Which name belongs to which class, something you have to mug up. Ascarius, Trichuris, Ankylostoma, Enterobius, strongyloids are examples of worm, and which worm is round, which is whip, which is thread, which is hook, which is pin, you should know. Then you have got platyhelminths, which are flat worms. They are flatty. They are flat worms. Example, you have got cestodes. Example is Taenia solium, Taenia saginata. And they have got a head called as scolex with the sucking organs and segmented body, but no alimentary canal. And they are tapeworms. They are flatworms, cestodes. Then you have got trematodes. They are non-segmented. They are non-segmented. You have to understand. Cestodes are segmented. with two suckers but no distinct head. They have now got no head. They have an elementary canal, and most important example is schizostoma and fasciolopus. Liver fluke and schizostoma is not leaf shaped. I hope it is very clear. So you have got nematodes, you have got cestodes, you have got trematodes. All the three are helminths. This is an example of heavy intestinal infections may occur with Ascaris. You can see a trichuris. You can see ankylostoma. You can see a strongyloids. You can see enterobius, which is pinworm or threadworm. Then you have got Ascaris roundworm. Examples of important metazoa, systemic nematodes, filaria. Then you have got Toxocara. You have got important cestodes. You have got intestinal cestodes. You have tapeworm, Tinea saginata, and Tinea solium. I have mentioned you regarding Tinea saginata and Tinea solium also. Tinea saginata and Tinea solium. Okay, Tinea solium is worldwide, acquired by ingestion of a contaminated uncooked pork. And Tinea saginata is by uncooked beef. Then you have got systemic Echinococcus granulosus. It's called as dog tapeworm. Then you have got Echinococcus multilocularis, which is called as a rodent tapeworm. Then these are your intestinal metazoans, tremodes, or flukes. This thing you can skip, maybe not very important for dentistry. Then you have got schistosomiasis. Schistosomiasis. Hookworm. Hookworm is Necator americanus and Ankylostoma duodenale. Hookworm causes anemia because it hooks around your colon and intestine and feeds on the blood. So the patient or the host becomes anemic. You can skip the life cycles and other things. These are your hookworms. So they try to move, they are motile and they try to move and hook up to your intestines. The heavy lukewarm infection results in chronic hemorrhage from a duodenal and jejunal mucosa, constant blood loss, and Ankylostoma duodenale. Hookworm. Lymphatic filariasis, I think everyone has been aware of Wucheria bancrofti and Wucheria malai. It's the female Anopheles mosquito again, popularly seen in African and Asian countries. Wucheria bancrofti is transmitted by Culex mosquitoes in India and Anopheline mosquitoes in Africa. The larval forms are called as microfilaria, taken up by a female mosquito when it takes a blood meal from a human infected with adult worms. The microfilaria develops inside the mosquito. When the mosquito takes another blood, the filaria from the mosquito's body will enter into the bite wound and enters the human circulation. Then they mature sexually in the adult forms over 12 months, depending upon the species of the filarial worm. So, who is the vector? Culex mosquito. Who is the host? Human. Who is the definitive host? Human. And then you can see Elephantiasis. You can see a swollen up feet, thigh, sometimes scrotum, massive swelling, and they lodge in the spermatic cords. Later stage disease is typified by Elephantiasis, painful, disfiguring, swelling of limbs. You can see an elderly male and you can see the Elephantiasis. You can see the nodules in the groin. You can see the enlarged scrotum. You can see a massive hydrocele. Lymphatic filariasis. And the drug of choice is DEC, diethylcarbamazine, rapidly kills the microfilaria. So this was a very brief introduction and a brief background of medical parasitology. Fine. Now we move on to the bacteriology, and then we start with streptococcal bacteria. Now we have already discussed about the shape and the structure of streptococcal bacteria. The bacteria is a cocci, and the bacteria is arranged in the form of chains. You have streptococcus. You have got streptococcus pyogenes, which is group A. You have got streptococcus agalaceti. You have got other hemolytic streptococci, viridans group streptococci, and you have got streptococcus pneumoniae. Streptococcus pyogenes leads to pharyngitis, scarlet fever, pyoderma, encephalitis, cellulitis, and you should know the names of these diseases because all these diseases, pharyngitis, scarlet fever, all of them are important streptococcal pyogenes diseases, and they're very important for you to understand. Scarlet fever, you can say as pyoderma, encephalitis, cellulitis, all of them, the whole family of these diseases are caused by Streptococcus pyogenes. Then you have got other Streptococcus bacteria. The classification is by Landsfield classification system based upon cell wall antigens into 20 groups. Then there is another classification based upon hemolysis reaction. You have got alpha hemolysis and beta hemolysis. Alpha hemolysis is Streptococcus pneumoniae, and others are collectively called as a viridans group of streptococci. Beta hemolysis is A, B, C, G, and some D. You have got a identification scheme: beta hemolytic colonies, bacitresin. You have got susceptible and resistant. You have a group A and group B. Streptococcus are gram-positive streptococci, ovoid cocci arranged in long chains, non-sporing form, non-motile form, can form capsules, and more parasitic forms are fastigious. Beta hemolytic is Streptococcus pyogenes. It is the most serious streptococcal pathogen. See now the question: which among the following species of Streptococcus is highly virulent? Answer is Streptococcus pyogenes. Streptococcus pyogenes is a beta hemolytic form of Streptococcus. In the microscopic, you can see specimens. You can see the cocci are arranged in the form of chain, typical chain. A Streptococcus bacteria, you can have a capsule, it's a gram positive. You have a cell membrane, you have a peptidoglycan cell wall. You know, it's a gram positive, so you know it is made up of lipoticoic acid, it is made up of ticoic acid, and so many things. So you know what is the basic nature of a streptococci. You know it very, very well. What does it do and what is the main reason it is. Beta hemolytic streptococci is the main representative of group A, most serious streptococcal pathogen. Strict parasite, inhabitates throat, nasopharynx, and skin. Produces surface antigens and virulence factors like M protein, C carbs, streptokinase, hyaluronidase, DNase, hemolysins, SLO, SLS proteins, and pyogenic toxins. Please remember the names of these enzymes: streptokinase, hyaluronidase, hemolysins, pyogenic toxin, key toxin development of scarlet fever. You know what is scarlet fever? Scarlet fever is again caused by Streptococcus pyogenes. It produces infection with tendency to spread locally along the lymphatics and throughout the bloodstream. C carbs, fibrate, these are all the virulence factors. Just the names, okay? Don't go in detail, just the names of them. The infections caused by Streptococcus pyogenes Superficially, it causes pharyngitis, skin soft tissue infection, erysipelas, impetigo, vaginitis, postpartum infection. Deep infections causes bacteremia, necrotizing fasciitis, deep tissue infections, cellulitis, myositis, pericarditis, meningitis, toxic mediated like toxic shock like syndrome, scarlatina, and then the most important, rheumatic fever, post-streptococcal glomerular nephritis, and reactive arthritis. These are the virulence factors of Streptococcus. As I mentioned to you, streptolysin, streptomycin, erythrogenic toxins, pyrogenic exotoxins. All of them are exotoxins because it's a gram-positive bacteria. Don't forget that. So the infections can be otitis, sinusitis, peritonsillar abscess, quinsy, suppurative cervical adenopathy, rheumatic fever, and post-streptococcal glomerular nephritis. It causes scarlet fever. Scarlet fever is important for us because you all are very much aware about strawberry tongue. I hope you all are aware of strawberry tongue. You have a soft palate, uvula, tonsil, everything is involved, manifested throat pain. You take a specimen from throat, respiratory infections like pharyngitis, tonsillitis, skin infections, superficial skin infections, impetigo, erysipelas. You can see the infection all around the foot. You can see how it spreads all along the foot. Remains superficial, but impetigo, you can see some here and there spots like lesions all over skin, but they're superficial. That's impetigo. Pyoderma, invasive group A streptococcal infections. This is called as toxic shock syndrome. Multi-organ system can become dangerous, can be involved. Flesh-eating bacteria, when it spreads, it can become to this level. Can you imagine? This is what is called as necrotizing fasciitis. It's a necrosis of the subcutaneous tissue, fat, muscles, and the fascia, and leads to a complication called as toxic shock syndrome, TSS. Invasive group A streptococcal flesh-eating syndrome or necrotizing fasciitis, again caused by pyogenes. Suppurative conditions like abscess in abdomen, brain, lungs, liver. Non-suppurative conditions like ARF, AGN, carditis, involvement of connective tissues of the heart. Rheumatic fever, delayed consequence of untreated upper respiratory infections. You know what is rheumatic fever? I think you have been thorough about rheumatic fever in general medicine. This is how it causes the valvular heart disease. I don't know if you know about something called as the vegetations, which happens. Then complications of streptococcal infection, acute rheumatic fever, scarlet fever, streptococcal toxic shock syndrome, glomerulonephritis, Andas syndrome. Then you have got post-streptococcal diseases, again rheumatic fever, rheumatic fever, sore knees, ankles, wrists, elbows, heart murmurs, hard to breathe, twitching movements, fever, sore throat, skin sores, all of them are rheumatic fever signs. And rheumatic fever is considered as a non-suppurative infection of Streptococcus pyogenes. Post-streptococcal glomerular nephritis, damage of the glomeruli, deposition of the antigen antibody complexes and complements inflammatory response. Then you have got streptococcal test, bacitracin disc test, rapid test to check the antigens. Treatment is definitely penicillin G or erythromycin and gram positive. Because it's gram positive cocci, don't get anxious seeing these things. It's okay. All that is not important. Then mutants, you know very well what mutants do. Caries and all, the all-time favorite of dentistry. This is also one of the basic classification of Streptococcus. I have simplified this. 3 classifications: Brown's, Lansfeld, and Griffith typing. You have got beta hemolytic, you have got alpha hemolytic, which is green. You have pneumonia and viridans. In viridans, you have got mutans, sanguis. In beta hemolytic, you have got pyogenes, group A, and agalacity, which is group B. Then you've got gamma hemolytic. Alpha, beta, gamma hemolytic is not very clear. Oval and commensals, non-sporing, non-motile, gram positive. Beta type of hemolysis. Ferment catalase negative is in red because you can differentiate it from Staphylo. Antigens, toxins, enzymes. M protein, T protein, R protein, then these are the names of toxins which I said to you. These are the names of the diseases. Respiratory issues, skin issues. This is also Syphilis, cellulitis, necrotizing, flesh-eating bacteria, genital infections, rheumatic fever, AGN. Now, this is a question of a 7-year-old girl with fever with severe sore throat, bilateral enlarged tonsils and exudates. What investigation will you do and what culture plate will you use to diagnose? Throat swab will you do? Yes. Direct gram staining, you see chains, gram positive. You take a transport medium, you can take a Pike's media or a Schwartz media. You culture on a blood agar. You go for ASO titer. You go for the bacterial culture. You check and you go for catalase negative. Similarly, 9-year-old girl, fever, sore throat, on throat culture, group A, streptococci, examination, pain, tender swelling, palpable effusion of right knee, culture of aspirate is negative, murmur is heard, diagnosis, you know what is it. Check for ASO titer. ASO titer is more, it is confirmed Streptococcus pyogenes. 7-year-old boy, 5 days later, colored urine, pitting edema. It can be rheumatic fever complication. Anyone sensitive to penicillin G will go for erythromycin. There's something called as the CAMP test. Just for your information, maybe I think once it was asked multiple, maybe many years back, CAMP test is done for which bacteria? The answer is Streptococcus bacteria. Or it is done to prove enhanced hemolytic activity. So all details are not necessary for you, but just you should be able to know. Viridans is a healthy group. It's like alpha hemolysis, mitis, mutans, salivarius, sanguis, all your common bacteria is viridans. Is it okay for all of you, or you need a break? You need a break, or everyone is fine with that? It is called as a pediatric autoimmune neuropsychiatric disorder. That is called as a panda syndrome. Panda syndrome is caused by streptococcal bacteria and it happens in children. And means basically it is a psychological problem. Basically, the children will get obsession. You may call it as an obsessive compulsive disorder, and which happens in children post streptococcal infection. Is it clear, Lakshmi? I am not taking continuous by a bacteriology because you will be confused because there are a lot of bacteria. Maybe we may not discuss all, but then few of them. So we are just discussing a little mycology, and then we'll again go back to bacteria. So mycology. Okay. We have the 4 major families of fungi, which is called as Zygomycetes, Ascomycetes, Basidiomycetes, and Deuteromycetes of fungi imperfecti. You have got molds like filamentous, penicillium, mushrooms, yeast, unicellular, you have got fission yeast and budding yeast. Then you have got dimorphic fungi. Then you have got yeast-like fungi. Molds, they grow as branching filaments called as hyphae. Hyphae forms a tangled mass called as mycelium. The growth happens at the tip of a hyphae and the vegetative growth from the fragments. This is how it is. You have a septal hyphae, they grow, they grow, they grow. When the hyphae will grow, they form a bunch called as mycelium. These are molds or mushrooms. If they have an asexual cycle, they grow from fragments. They have a pores, asexual spores. called as blastospores, arthrospores, chlamydospores. If it is from sexual cycle, it is very rare, less frequent. There'll be fusion of nuclei like oospore, zygospore, ascospore. The very important question is the lab diagnosis of any fungal infection happens in 10% potassium hydroxide preparation. You write it, put a star mark to that point. Potassium hydroxide preparation is an ideal staining preparation done for the diagnosis of fungi. Gram staining done rarely, but they show gram positive. Sometimes you have to use an India ink preparation, which I mentioned you before, is a negative staining for capsulated bacteria or viruses or fungi, capsulated yeast like Streptococcus neoformans. And I have also mentioned you yesterday the culture medias for fungi, and I've told you that the only two culture medias for fungi is Severod's dextrose agar SDA media and brain heart infusion SDA and brain heart infusion media. Very important, all of you should know that. You classify the fungal diseases. The fungal diseases are broadly named as mycosis into superficial mycosis, subcutaneous mycosis, and systemic mycosis. You have got superficial mycosis, the superficial mycosis, which you know, what is it? The systemic mycosis. So superficial mycosis is nothing but happens on skin, hair, nail, mucus. Can be on surface. You may call it as dermophytes, tinea nigra, or versicolor. Dermatophytosis. They utilize only the keratin, doesn't penetrate into subcutaneous infections or tissues. Classic ringworm infection. Ringworm is a fungus and basically is a superficial mycosis. Subcutaneous mycosis is mycetoma madura foot. You all are aware of it. It is also called as chromomycosis, porotrichosis, non-specific ulcer in the oronasal pharynx. Rhinosporidiosis is a soft red polyps in the nasal mucosa. So means why am I telling you this? Like 444, because if someone asks you which among the following is not a subcutaneous mycosis, you should know that Madura mycosis, chromomycosis, porotrichosis, and rhinospor, all the four MCSR belong to subcutaneous mycosis. Similarly, dermatophytosis belong to superficial mycosis. Systemic mycosis, very important, histoplasmosis, because it is related to oral cavity. It causes nodulo-ulcerative lesions in the oral cavity. It is called as histoplasmosis. Blastomycosis, you have got North American blastomycosis, South American blastomycosis again. You have got paracoccidomycosis and cryptococcus, which is seen in HIV. Then you have got name of the four very popular opportunistic mycosis. You have got Candidiasis, you have got aspergillosis, you have got mucormycosis and phycomycosis. This is important that pneumonia in immunocompromised patients is caused by pneumocystis jirovecii. Move on to oral candidiasis. Actually, this will be covered in detail in oral pathology. But if you want, I will talk about it, or I can skip. Just let me know your vote if you want me to talk about it. You want me to talk about oral candidiasis? Yes. Okay. Okay. Oral candidiasis. It is called as candidosis, or also maybe previously called as moniliasis. It can be of various forms, various classification system. Acute candidiasis is pseudomembranous candidiasis and atrophic candidiasis. You have got pseudomembranous candidiasis and atrophic candidiasis. Example, antibiotic stomatitis. Chronic candidiasis, you have got again atrophic candidiasis, hypertrophic candidiasis, and multifocal candidiasis. So acute pseudomembranous candidiasis is very popular. Under acute atrophic candidiasis, it is called as antibiotic stomatitis. You may call it as antibiotic sore mouth. In chronic, you have got atrophic and hypertrophic. In chronic atrophic, you have got denture sore mouth, angular cheilitis, and median rhomboid glossitis. Under hypertrophic candidiasis, it is candidial leukoplakia, papillary hyperplasia of the palate, and median rhomboid glossitis. Then you have got the mucocutaneous candidiasis. You have got syndrome associated familial or endocrine candidiasis syndrome. Oral thrush, the superficial infection of outer layers of epithelium, patchy white plaque or flecks. You know what is oral candidiasis, oral thrush, right? Scrapable lesions in female, children, or adults. Scrapable like a cottage cheese appearance. Typical lesions are patchy, painless, and when you remove them, you see an underlying reddish erythema. Infant thrush. Any mucosal surface may be involved. Bad taste, loss of taste discrimination, burning sensation. Then you see a cottage cheese or curdled milk appearance. White patches are wiped out with a wet gauze, which leaves a normal or an erythematous area or an atrophic area behind at the base. The organisms involved mostly are Candida albicans, tropicalis, and glabrata, account for about 80% of the medical isolates. The rare one is Candida, and other species are there. Predisposing factor can be because of huge or a over-the-counter usage of antibiotics, especially broad spectrum, sometimes Cetra or amoxicillin or maybe penicillins, excessive use of antibacterial mouth rinses, chronic local irritants, administration of steroids, inhalers, nebulizers, including steroids, pregnancy, immunologic deficiencies, xerostomia, chronic local irritants. The differential diagnosis for candidiasis is plaque form of lichen planus, leukoplakia, genodermatosis, gangrenous stomatitis, chemical burns, acute atrophic candidiasis, acute atrophic candidiasis, antibiotic sore mouth, chemical burns, drug reactions, syphilitic mucosal patches. Then you have chronic atrophic candidiasis, which includes denture stomatitis, angular cheilitis, median rhomboid glossitis. So there is a little confusion, like antibiotic sore mouth is acute. Means mostly they ask you because acute sore mouth and chronic sore mouth are different aspects. Acute sore mouth is acute candidiasis caused by antibiotics. Chronic sore mouth is caused by dentures. So chronic sore mouth is nothing but denture sore mouth, which is also called as chronic atrophic candidiasis. So be aware of the distinction between both the candidiasis. Then you have got angular cheilitis and median rhomboid glossitis. Then you have got denture stomatitis or a denture sore mouth, which is chronic atrophic candidiasis. Long-term dentures, poor hygiene, not removing it, not cleaning it. First you get palatal petechiae, then you get diffuse erythema, and then you get development of tissue granulation, papillary hyperplasia. And chronic atrophic candidiasis, denture sore mouth, rarely found under metabolic denture. Sorry, mandibular denture. Angular cheilitis. There is frequently a coexistent denture stomatitis and uncommon in patients with natural dentition. Other possible etiological factors include these: lip commissures, angular cheilitis, then median rhomboid glossitis, chronic hyperplastic candidiasis. It can be Candidiasis, chronic multifocal candidiasis, chronic mucocutaneous candidiasis, immunocompromised associated candidiasis, and Staphylococcus. You have positive cocci, bunch of grapes, they look like berries, pyogenic infection, pigment production not associated with virulence. Again, this is very important. Golden yellow colonies by Staphylococcus aureus. White colonies is Staph epidermidis or albus, and the lemon yellow colonies is by Staph citrus. Lemon citrus, so you can remember that. Staph, they're like bunch of grapes. Non-motile, non-sporing, uniformly positive, L forms may be seen. I mentioned you before also. Coagulase positive is Staph aureus. Coagulase negative is the various other bacteria. Culture media, aerobes, nutrient agar,
Sir,
slightly yellow
slide
colonies.
is not visible.
Not visible?
No, sir.
Okay. Now visible?
Yes, sir.
Okay, okay. I'm just sorry. I'll just repeat the slides. You cannot have you not seen anything from Staphylococcus? Staphylococcus totally you have not seen any slides? Okay, 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 okay. Now you can see all right. Okay. Staphylococcus gram-positive cocci, as mentioned you. These are the slides. These are the cocci bacteria, and I was talking to you regarding the colonies. I was talking to you regarding Streptococcus. Sorry, Staphylococcus aureus is golden yellow. Lemon yellow is Staph citrus. You have white colonies, which is Staph epidermidis. You have got bunch of grapes. Coagulase positive, Staph aureus. Catalase positive. I have told you. Catalase negative is Strepto. Catalase positive is Staphylo. So if the patient is resistant to your penicillins, or maybe also includes beta-lactamase resistance, then you may have to go for methicillins or cloxacillins. Sometimes patients are resistant to these also, then they are called as MRSA species. You may go for oxacillin. Resistant to that, you can go for cefosaxin. Pathogenicity, they enter the damaged skin, mucosa, tissue damage, intoxications with the bacterial toxins. They are gram positive, so definitely there'll be peptidoglycan, there'll be teichoic acid, there'll be antigens, there'll be a capsular polysaccharide, there'll be a good amount of virulence. You'll have a protein A, you'll have a clumping factor, you have an extracellular enzymes. You have a coagulase test. Violence factors, lipases, hyaluronidase, nuclease, protein receptors. You have got toxins like cytolytic toxins, alpha hemolysins, beta hemolysins, gamma hemolysins. Super antigens. It leads to toxic shock syndrome toxin. It's a multisystem disease. You see a toxic shock syndrome toxins antibodies. It leads to SSSS Staphylococcal scalded skin syndrome. It is an exfoliative syndrome, epidermolytic syndrome disease. And in children, it is called as Reiter's disease. You can say Ritter's disease in children. In older patients, it is called as toxic epidermal necrolysis. Please take a picture of this slide. This is very important. It's a toxic mediated syndrome. You can see the skin changes. Staphylococcal disease, skin soft tissue infections, folliculitis, furuncles. So all your small lesions on skin like carbuncle, furuncle, boil, abscess, impetigo belong to staph. Impetigo goes to strepto also, but basically boil, carbuncle, furuncle, folliculitis, sty are all staphylo. Most of the diseases, lesions of staphylo are localized, restricted on skin, on mucosa, purulent, pus discharging from the lesion on the skin or wherever you can see it very clearly. Whereas when you see strepto, you don't see localized lesion, you see a spreading lesion. I have mentioned you, Erysipelas, you saw the lesion is there on the calf, but the swelling you have seen in the foot. Why? Because all the streptococcal bacteria which are beta hemolytic in nature, like Streptococcus pyogenes, release streptokinase and hyaluronidase. That is how it is very informative for you to know that streptococcal infections are not in one place. They have a capability of spreading into subcutaneous tissue and becoming. That is why strepto is more virulent and intoxicating compared to staphylo. Staphylo infections are restricted to a place of point of entry, purulent, involving the epidermis, the superficial mucosa. Strepto erodes, invades, spreads, and destroys the underlying tissues, the connection barriers between the muscles, the skin, and causes cellulitis. In fact, this doesn't cause cellulitis. This causes local lesion, local ulcer, local pus. And I think you should be very clear with those things. Staphylococcal diseases, same thing. Both strepto and staphylo commonly can cause respiratory infections. But then, because both of them are gram-positive cocci, the line of management remains again the penicillin. And then if I say, how do you differentiate both of them? You know very well. The structure, the shape, the biochemical reaction. Penicillin's drug of choice, if it is MRSA, then vancomycin. Vancomycin, teicoplanin, resistant strains have appeared now. Superficial infections, local applications with bacitracins are there. There are some staph which are coagulase negative also. There are few of them which are coagulase negative, like staph epidermidis. This is commensal on skin. Staph hemolyticus, staph saprophyticus. Okay, so we take a break for 5 minutes. Is it okay for all of you? Can we take a break for 5 minutes? Can I start? We'll start with Vibrio cholera. We can call it as Vibrio cholerae. And cholera, you know very well, it affects millions of people in endemic areas and causes thousands of deaths, especially in seasonal epidemics. Robert Koch was the first microbiologist of Germany who has discovered Vibrio cholerae because of its characteristic feature of a comma-shaped bacilli. It spreads from water and food. It's a comma-shaped curved rod, 2 to 4 micrometers long, actively mobile by means of a polar flagellum. On a prolonged cultivation, Vibrios may become straight rods and resemble a gram-negative rod. Vibrio has got various species, but Vibrio cholerae has got non-O1 and O1. And contaminated water and food, raw, undercooked, or uncooked seafood. Person-to-person contact is rare, but fecal-oral transmission is highest. And I said, and you all should know, the name is quite maybe surprising for you. An Indian name, Venkatraman Ramakrishnan medium. The simple medium which is used as a transport medium, and you can use it to be cultured in an enrichment medium. I told you enrichment mediums are liquid mediums. I have mentioned you before also. You can call them as a broth. They are done on alkaline peptone water. Vibrio cholerae produces convex, smooth, round colonies. They grow well on a very higher pH and are rapidly killed by the acid. Vibrio cholerae grows well on thiosulfate, citrate, bile, sucrose, agar. Growth characteristics: ferment sucrose and mannose, but not arabinose. No need to go in detail with the antigen groups of Vibrio cholerae. Remember that what is concerned for us, the highest virulent is Vibrio cholerae O1. That is the serotype we are concerned with, responsible for causing the epidemic of cholera. So you know what happens? Decolonization, hypovolemic shock, vomiting, severe diarrhea, cramps, and acidosis. It's not invasive. It doesn't go to blood. Affects locally into the gastrointestinal system. Multiply, release cholera toxin, and perhaps mucinase and endotoxins. Diarrhea is 20 to 30 liters per day, you can imagine. Absolute shock, hypovolemia, hypovolemic shock, that can lead to death. 60% is caused by classic Vibrio cholera issues, but 75% caused by other biotypes also. Incubation period 1 to 4 days, manifest with nausea, vomiting, diarrhea, cramps, rice, water, stool. Characteristic, please remember or underline, make some notes or somewhere. Rice, water, stool is the pathognomonic feature of cholera. Loss of fluids, profound dehydration, circulatory collapse, hypovolemic shock, anuria, because there's nothing to flush out. Everything is coming out as a dehydration. The patient will go into oliguria and anuria. Rice water diarrhea, the sample picture. Stool specimens, you see so many choleras. You may see in your exam also a picture, and they may tell you a patient came with this report, and this is a specimen with that, this, and all, blah, blah, blah. And the answer is Vibrio cholera. Stool specimens are collected in acute stage. Before antibiotics are administered. And you use to carry Blair's transport media. You have to use, as I mentioned, you V cholerae O10 group serotype. You have to confirm the cholera toxins. Cholera rapid test, dipstick test. Sometimes you see mucus flecks from stools. Dark field microscopy, as I mentioned, you will show the motility of the bacteria. You can go for Venkatramana Ramakrishnan medium. Alkaline peptone water is the medium of choice for growing it. And you may see the agglutination reactions, immunity about it. So patients will suffer from diarrhea only, preferably. First symptom is diarrhea only. Treat all cases, but suspected cases, you need to collect the sample, check for the specimen, bacterial shape, culture. ORS, oral tetracycline. Maybe you can go for vaccines for cholera. Is basically people have got a herd immunity for it. So basically, very rarely you hear about cholera today in the global situation. So vaccine is no longer required as such. But anyhow, there are two types of vaccines for cholera: Dukoral vaccine and Shankol vaccine. If you wish to know more detail about cholera, maybe if you have keen interest into it, you can go and refer these guidelines for cholera control. Tuberculosis. Obligate aerobes growing more successfully in a higher oxygen content organs like lung. Facultative intracellular pathogen, usually affecting the mononuclear phagocytes, which are macrophages. You've got a very wide classification range of mycobacterial tuberculosis, mycobacterial leprae, mycobacterial bacilli, causing skin ulcers, causing atypical mycobacteria. Mycobacterium differs from regular, routine, isolated bacteria, slow growing with generation time of 12 to 18 hours, hydrophobic in nature, acid fast bacilli. This is very important. I have already told you what is acid fast bacilli or acid fast staining. How their acid fast is once stained, the cells resist decolorization with acidified organic solvents, and therefore the acid fast in nature. Mycobacterium tuberculosis complex. Mycobacterium bovis. What are atypical mycobacterium is infect birds, cold-blooded animals, worms. You know what is TB, how it affects the lungs. Has infected very, very popular people. Obligate aerobes, I have already told you. Slender rods, acid fast staining, acid fast bacilli. Why is it acid fast? That's because of unsapnofiable wax, mycolic acid, which is a semi-permeable content of a cell wall of this bacteria. That is why they are acid fast. You know why gram stainings? Because of peptidoglycan. So why are they acid fast? Is because of mycolic acid content in the cell wall. Cultural characters. Culturing acid fast bacilli. And I think, I don't know if you remember, the media for tuberculosis is Lernston-Jensen media. Always remember the name of the media. It's Lernston-Jensen media for your awareness. You should be thorough about this bacteria. It contains coagulated egg, one of the very, very important mediums. Not routinely used, but other mediums is Middlebrook media and Sula's medium. On LJ medium, you can see appears dry, rough, raised irregular colonies. They appear creamy white, yellowish. Lernston-Jensen media. On liquid medium, appear as long as serpentine cords in liquid medium. You know how it spreads. You know how it spreads by respiratory route, droplet infection, attacks the lung, the effects of the lung, at the area rich of alveoli where there's a lot of oxygen. Forms, I think you all know what is Ghon's nuclei and Ghon's complex. Generation of droplets, inhalation, Granulomatous is disease. Granulomas in the lung tissue. They don't produce any toxin, but they multiply. They form granulomas, which is called as tuberculoid. Inside has got caseous necrosis. You can see T cells. You can see epithelioid cells. You can see giant cells. You can see caseous necrosis. You can see tubercle discharging out of it. You've got a hard tubercle. You've got a soft tubercle. And you know about the yesterday mentioning you about the delayed hypersensitivity reaction, Th2 manifestation. Center, you see caseation or caseous necrosis, cheese-like necrosis, exudative. Primary happens in an endemic without any reason. You know about Gons focus, Gons complex, primary complex. Gons focus with enlarged lymph nodes appear after 3 to 8 weeks. Heals in 2 to 6 months calcified. Reactivation of tuberculosis. Coach phenomena. Very important. There is a question. Coach phenomena associated with tuberculosis. Bacilli. Post-primary tuberculosis spread is more. It goes to miliary tuberculosis, systemic tuberculosis, meningitis, pleurisy, kidney, spine. It's called Pott's spine. Very important question will be discussed in general surgery. What is Pott's spine? Pott's spine is nothing but tuberculosis of spine. You know the most important symptoms of tuberculosis. Sputum smear stained by Zeal-Nielsen staining, which I told you, acid pass staining or Zeal-Nielsen staining, whatever you feel like. Then culturing, LJ medium, tuberculin test. Then you have got BATEC 460 rapid radiometric culture system test, PCR. It happens very common with HIV because of immunosuppression. First line drugs, you all know very well. Second line also you know very well. BCG full form, intradermal injection of live attenuated vaccine. Clostridium tetany. Clostridium contains a large gram-positive spore-forming species which produce and affects humans. Clostridium tetany is a rod-shaped anaerobic bacterium. anaerobic bacterium. Gram positive, drumstick appearance, obligatory anaerobe. Grows on blood agar with advancing edges. And I said you before, Robertson cooked meat because it is anaerobic, obligatory anaerobic. Spore forming, drumstick appearance. Tetanus, you know very well, entry and source and skin and all those things. You know very well all these things. You will read in depth in general surgery. Traumatic tetanus, C tetanus, tetanolysin, tetanospasmin. You know what is the various things that happens with tetanus is hemolysin, neurotoxin, non-spasmogenic. Tetanospasm, you know, you heard of Rhesus sardonicus, R-shaped back. Yeah, you get the lockjaw. The neurotoxins are there. It leads to permanent contraction. Opisthotonus, R-shaped back. Tetanus symptoms, Rhesus sardonicus, opisthotonus. Characteristic feature, a rigid smile, trismus, lockjaw, rigid arc back, opisthotonus and Rhesus sardonicus. Classic features of tetanus. Drumstick or tennis racket appearance. Spore which is terminal. Robertson cooked meat agar. Remember the media's name. Treatment is especially metronidazole, or you have got TT, toxoids. You have got ready-made antibodies for this. You have passive immunization. You have active immunization. Tetanus toxoid. Passive immunization. You have got anti-tetanus serum, which is made from equine. You have anti-tetanus serum human. Passive immunization. You have ready-made anti-tetanus serum equine, or maybe from human also sometimes we get. Conibacterium diphtheriae, gram-positive, aerobic rod, non-acid fast, non-motile, club-shaped swelling at one or both ends, so name Cornybacterium, especially leading to sore throat. Very contagious, life-threatening infection when goes down into respiratory tract, attacks the throat, club end, non-sporing, non-acid fast, non-motile, toxoid made from diphtheria toxin. Has three serotypes: Corynebacterium, diphtheria gravis, intermedius, and mitis. Local lesions and pathogenesis. It affects the fauces, the faucial pillar, the tonsillar pillars. Typical presentation of bull neck, nasal diphtheria, faucial diphtheria, local manifestation. You can see how it leads to faucial pillars, Corynebacterium diphtheria, slender rods, clubbing at both the ends, pleomorphic. Granules are composed of polymetaphosphate and stains with leuphulose, methylene blue. Chinese letter pattern. Stain is Albert stain. Leuphoserum slope or telluride blood agar. Gram positive, aerobic bacteria, rod, non-motile, non-culturing. The Leuphoserum slope grows rapidly in 6 to 8 hours, forms white disc. Commonly used media, telluride blood agar, gravis, intermedius, and mitis. Pathogenicity, diphtheria, starts from malice, sore throat, fever, pseudomembranous slowing of the what do I say? The oropharynx goes down to the larynx. You collect the thick membrane swab culture, blocks the whole throat, sometimes skin lesion, faucial diphtheria, septic ulcerations of your intestines, of your what do I say? The viscerals, leading to asphyxia because of the thickness of the membrane, respiratory blockage, asphyxia, mini tracheostomy, post diphtheria paralysis. Bacteria may produce endocarditis, meningitis, basically pharyngeal diphtheria with pseudomembranous obstructions, fever, peritonitis, isolation, swabbing, toxigenicity, throat swab, Albert stain, Leuphoserum slope, telluride blood agar, Schick test, but outdated, but Schick test is done. Immunization, active, passive, both passive and active. Active immunization, prevention, passive immunization. Antibiotic not useful in acute infection. You heard of diphtheroids. They resemble Corynebacterium diphtheria. They are also commensals in throat and skin. Example is Corynebacterium hoffmanni and xerosi. They are arranged in parallel rows, unlike diphtheria in Chinese letter pattern, lack metachromatic granules. They have metachromatic granules. The last bacteria is spirochetes. We're coming to the conclusion with spirochetes. They are elongated, motile, flexible bacteria. They contain endoflagella. Family is Spirochetaceae. Genus is Treponema and Borrelia. It can be also Leptospiraceae or Leptospirea. Treponema, Borrelia or Leptospira. Borrelia, Treponema. We have got Treponema pallidum, endemic syphilis. Spirochete, motile, sluggish, quite long, endoflagella, and you know that you need to have dark field microscopy for that. Spirochetales is the order. They are gram negative, appear like coiled hair, maintained in anaerobic circulation, medium with albumin sodium. Endoflagella, stains by Giemsa stain and Fontana stain. Take a picture of the slide. Staining with special stains like Giemsa and Fontana. Again, important slide, the test for syphilis, Wasserman, Hahn test, VDRL test, beef heart extracts antigen, lipid hapten, cardiolipin. This is dark field microscopy. Treponemes are distinctly related to the gram-negative bacteria. Primary syphilis is called as canker. You see on genital organs. They are canker. And you see a latent disease. It can be oral or genital. Secondary syphilis. In secondary syphilis, you see snail track ulcers on the lateral surface of the tongue. You see something called as a mucus patches. which are the most contagious stage of syphilis. Then third stage is gamma. You see neurosyphilis. You see pustules everywhere. Congenital syphilis, you all are very much aware of it. Saddle nose, Hutchinson's teeth, interstitial keratosis, eighth nerve deafness, mulberry molar. Mucocutaneous rash, as you see here. Highly contagious form, as I said to you. You see rashes like this all over the body. Gammatas, rubbery tumors all over the body in tertiary syphilis. Gammas are localized granulomatous lesions. Neuros, latent, congenital transmission to child or the fetus. You are aware of Hutchinson's triad, dark field microscopy, silver staining, because I told you that you need to do silver impregnation for thin bacteria or flagella. Serological test, most important is VDRL. Serological testing, treponemal, non-treponemal testings. Serological test, and the antigen is called as cardiolipin and lecithin. VDRL, RPR, fluorescent treponemal antibody absorption test, serological test, it's too detailed. Basically, I prepared for medical students, so it's too much for you all. But just basics. What is the other treponemal diseases names? If you know what is yours, Pinta, Beagle. The last is a little bit about virology. You know viruses, you are very much aware of it. You are hearing since a year about viruses and viruses. You know what is DNA, RNA. The smallest is the parvovirus. The largest is the smallpox virus. They are resistant to some disinfectants. They are resistant to most of the sterilization aspects of it. You know what is a capsid? You all know about the virus has got a capsid, which is made up of capsomers. Inside you have a nucleoprotein. DNA or RNA plus capsid is called the nucleocapsid, which is a naked capsid, sometimes enveloped, the envelope virus. You have a virus capsid symmetry, can be isocohedral, helical, and sometimes can be an enveloped virus. This is a naked virus, could be a helical virus. And it's absolutely sometimes resistant to most of it. Naked are resistant to some of this. Very well aware that viruses can be cultivated in living organisms only into an animal or an embryonated egg or a tissue culture, but not on culture plates, broths, agars, media, nothing. It needs only these three. And it's an important question. Which among the following is not a method of employing virus cultivation? 1, 2, 3, and 4 will be the answer. The detection of the virus growth in the cell culture. Classification of virus into DNA, RNA viruses based upon the nucleic acid. Every virus has got a nucleocapsid. You have a DNA virus. You have a pox virus. You have a herpes virus family. You have an adenovirus family. You have a papovavirus family. Pox is the brick-shaped, large one, smallpox, largest. Variola virus called smallpox. Then you are aware of herpes. In herpes, you have alpha herpes viridae, beta herpes viridae, gamma herpes viridae. Alpha groups belongs to herpes simplex 1, 2, and VZV. Beta causes human cytomegalovirus, CMV, with herpes 6 and 7. And gamma contains EBV and herpes 8. Be aware of this. Take a photograph of this slide. You should be very familiar with the classification of herpes virus. Herpes labialis, Zang cells and Cowdery type A bodies, very important points. Herpes can be differentiated by Zang cells and Cowdery type A inclusion bodies. These are the bodies, or these are the inclusion bodies which you see in the virus affected cells. This is what is the bodies. VZV, varicella zoster virus, causing chickenpox, adult herpes zoster. CMV, important in AIDS, very latent cytomegalic cells called as owl's eye cell. Epstein-Barr, actually called as herpes virus 4, but belongs to gamma herpes viridae. It causes infectious mononucleosis. It causes nasopharyngeal carcinoma, Burkitt's lymphoma, Hodgkin's lymphoma. With HIV, a lot of things, and autoimmune also. Very, very important virus, HHV4. Infectious mononucleosis, inclusion bodies, megabodies, they are called as megabodies. Adenovirus causes respiratory issues, genital issues. Papovirus, oral lesions, oral papillomavirus, you know very well, papillomatosis, human papillomavirus, genital warts. HPV is a DNA, is the only hepatitis virus which is DNA, is hepatitis B. Absolute long incubation period, but affects because of a lot of reasons which you are aware of, same like HIV. And you can treat by passive immunity and active. And whenever we talk about active immunity, go for vaccine. Passive immunity is ready-made immunization, anti-serum, antibodies, toxoids are all passive. RNA is picornavirus. You have your polio virus, Coxsackie virus, hepatitis A virus, polio. You know what is polio? Immunoprophylaxis, skilled Salk and live agent Sabin, Salk and Sabin virus. Vaccines are there for rabies, sorry, for polio, sorry. Then you have orthomyxovirus, it's influenza virus. Paramyxovirus, you have parainfluenza, measles virus, and mumps virus. Then you have rabies virus. You have arbovirus. Arbovirus is your dengue virus. Retrovirus is HIV. HIV-1, HIV-2. HIV-1 is worldwide, 2 is West Africa. So a lot of time takes for incubation of HIV-2. Commonly what we see is HIV-1. You know what is HIV, you know the CDC stages of HIV, acute HIV, asymptomatic lymphadenopathy, and full-blown AIDS. Human T-cell lymphotrophic virus, STLV virus. Again, retrovirus, same class of HIV-1. Hepatitis C is bloodborne pathogen. You know what is prions? It's an infectious proteins without nucleic acid. They're not viruses, but they lack nucleic acid. Maybe if you have heard of Creutzfeldt-Jakob disease, I think I mentioned you in Perio, if you remember, during bone grafting chapter when we were discussing about bone grafting. In animals, it is called as bovine spongiform encephalopathy, mad cow disease. In humans, it's called as Creutzfeldt-Jakob disease. I told you about bacteriophages, transduction, tadpole shape, bacteria to bacteria transduction. There's something called as the prophage. It's the genome which is phage DNA plus bacterial DNA. It's called prophage. Means the virus DNA with bacterial DNA, when they fuse, it is called as prophage. Then you have a lysogenic bacteria, the bacteria with prophage genome. And that completes virology. Yeah. Any doubts? A national immunization schedule? No, I am not aware of the latest guidelines. Silver staining is done for spirochetes. Yes, it is done for spirochetes. Anything which you are not able to see, you have to silver staining. See, basically, tuberculin test is something where you inject the bacteria into the subcutaneous region of your arm and you see the reaction of that. That is what is tuberculin test. Means it's a positive or negative. If you see the reaction about the tuberculin test, then you will come to know that the bacteria has already attacked. That is what happens. The tuberculin test is basically done. And I think if you know very well that tuberculin test is also called as Montauk's test. It is basically used for screen tuberculosis. Did you understand? Yeah, Montauk's test and tuberculin test is the same. Tuberculin test is also called as Montauk's test, where you inject the bacterial antigen and you see the reaction. If you are already exposed to tuberculosis, you will get a reaction on your skin. Okay. If you're already exposed to the tuberculosis before, the antibodies will fight with the antigen and you see a red color small lesion on the arm. That is there. Okay, that is tuberculoid test. Is it okay? Who asked me? Lakshmi has asked me, right? Negri bodies are nothing but they are small, inclusion bodies, and you see those bodies in the cells affected by rabies virus. Negri bodies are inclusion bodies. There are very few inclusion bodies named which you must remember, like Negri's bodies for rabies, mega bodies for infectious mononucleosis, Cowdery bodies for herpes. Yeah, anything else? I didn't get you. What part of immunity means? I don't know, ma'am. You can ask someone else. I have no. I'm not aware of that. I don't think so. I can answer that. I don't know whether the question is also relevant or no. I don't know that also. How much I know, it is CMI, but I'm not sure. I think it is CMI because tuberculosis is caused by type 4 reaction. Type 4 reaction is a CMI reaction. It's a delayed hypersensitivity reaction. It's a T cell lesion, not a B cell lesion. Is my knowledge says. But maybe if you have someone senior to me, you can definitely ask. You can ask the oral pathologist. Maybe they are more aware of it. Maybe you have oral pathology and general surgery classes, right? You can ask some surgeon if they can tell you because they are more experienced with the lesions. Is it okay? Shall I end the meeting? Shall I end the meeting? Okay. Fine. With this, we complete microbiology. So I'll just brush up with all the topics we have covered with independent, little bit about bacteria, little microbiology, little virology, little parasitology, mycology, some media's name, some examples name, some diseases, toxins name, some media's name. If you remember, that's more than sufficient. Don't break your head reading in detail about the antigen reactions, the pathogenicity. There's some diseases, some classic features like diphtheria, sloughing, like cholera, rise water stools, like streptococcus, something else, rheumatic fever, post-pneumonephritis. Read something which is very important and star points of microbiology. Some media's name, Venkatramana media, Leuphosserum slope media, Robertson media, spirochetes, silver staining, impregnation methods, VDRL. Read all those things. Then viruses, HIV1, HIV2, Seborrheic dextrose agar, QOH preparation, and where do you grow virus, herpes virus in detail. No need to go and read what is pox virus, what is adenovirus, what is parvovirus. All that is not important. So unnecessarily don't read extra, but read everything. But just like I always say, right? You should know uh, something about everything. That's how it is. If you want to know everything about everything, then maybe you can write exam in 2021, not in 20. It's very difficult. So, and there is no end to knowledge. So, try to know little about everything. Okay. So, I think I will meet you for the last session of biochemistry. For the next, maybe one of the for your batch, the next subject I will be taking will be the biochemistry. And biochemistry, we will do it again the same way, as brief as we can, because I know it is one of the most driest subject. That is why I kept it in the last. But then in biochemistry, anything specific you want me to teach, then I would request you to convey it to me, because biochemistry is something which is not cup of everyone's tea. So if you are in specific need of biochemistry, so instead of asking me doubt, it's better you ask me today or tomorrow what exactly you want me to teach you in biochemistry. I can schedule the presentations accordingly, because I cannot talk about every topic, all the subjects I have been talking about every chapter. But biochemistry, we cannot do every chapter because it is not important. The weightage of biochemistry is not more than 1 or 2 questions in the total paper. So what you want me to teach, focused teaching, and without wasting my time and your time, if you can tell me, I can focus on that. So I hope and wish that you can convey it to me through Dr. Ahmed, and just let me know what you want. Okay, fine then. Thank you so much.